So we have an activity planned for you guys to go through and actually get to do some of the design aspects of this. And you'll be actually designing a cassette. There's 6 parts of a cassette, and each part is really important. And this cassette is the actual sequence that you would have synthesized and then put it into your organism. And each piece is important for a different reason. You start with the promoter, which is your first piece. And it's needed to turn on the gene. It promotes transcription, which is that process from DNA to RNA. It's going to start that process. It's specific to the type of organism. So there's bacterial ones, there's plant promoters, there's animal promoters. So you have to be really careful about which type of organism you're working with and make sure you pick the right kind of promoter. And they also have various levels of strength. People have categorized the strength of them, so some of them will better produce the proteins that you want to make with your gene sequence. The next part is a binding site. These are needed for translation. It's what helps the ribosome know where it needs to latch on to that RNA sequence or that RNA piece. And so it's needed to start the process of putting those amino acids together to get your protein. And there's prokaryotic ones, so your bacterial ribosome binding sites are called a ribosome binding site. And the eukaryotic ones, so all your plants and animals and all that stuff, that is called a 5' cap and eukaryotic initiation factor. They're just different depending on the organism. Then you have your start codon. It will always be AUG. And it's found at the beginning of the gene of interest. It is what tells the ribosome that it's ready to start that amino acid chain. So it signals the ribosome to go get the methionine, which will be the first amino acid in any protein that is made. And then you have your coding sequence, which is the important one. It's the one that codes for whatever trait you're trying to get expressed. They're typically very long. A lot of them are, at least. And they're the cool part of the cassette, I guess you could say. And then you have your stop codon, which follows the coding sequence immediately. And there's 3 different stop codons. They're all the same in their strength and effectiveness. But it's what signals the ribosome to let that amino acid chain detach, and you'll end up with your protein. And then you have your terminator sequence, which signals the end of transcription. And it's going to follow your stop codon to make sure that your stop codon gets transcribed before transcription is over. So those are all the pieces. It's really important to have them in the right order. If they're in the wrong order, it's not going to work. If you put it into an organism, the process of going from that DNA to your protein is going to fail, and you're not going to see any results. So it's really important you have them in the right order, and you have all the pieces specific to the organism that you're working with, because if they're not specific to that organism, again, the organism won't recognize it, and it won't allow it to go through this process to make your protein that you need. So we're going to have an activity. Do you guys want to come help people? All right, so we have an activity, and you're going to get into 4 groups. You guys can decide them yourselves. But you're going to get into 4 groups, and you're essentially going to be a team. You're a team of biologists, and you're going to be designing a cassette. So 2 separate companies are going to come to you, well, on a piece of paper, and tell you what they need. They're coming up with new products to make their business better. And so you're going to not only have to take into account all the scientific things that need to go on to make sure you have everything in the right order and that kind of stuff, but you're also going to try and make a product that's going to be really marketable and really desirable to consumers. So there's 2 different scenarios. We'll do the first one, and then we'll do the second one. You're going to get into your groups, and you're going to have a certain amount of time. We'll see how long it takes. And then you're going to explain why you did what you did. Science is all about explaining what you're doing and why it's being done and why you think it's going to work. And so you're going to need to have a reason for why you made your cassette the way that you did. Okay, are we good? All right, if I could just have your attention, I'm going to explain what you guys have here. So you're each team is going to get this kit thing and a whiteboard. And if you want to write on your whiteboard, it's fine. But inside, you're going to have the 2 companies and what they're looking for. And we're just gonna do them separately, so don't look at the second one yet. We'll go over the first one. And then this is a list of all the pieces that you have in the kit. So in the kit, you're gonna see all these different pieces. They're little foam pieces with magnets on the back. And on them, they say what they are. And then on here is a little bit more of a description about what they're for and all that kind of stuff. And so you're just gonna go through and put on your whiteboard, just put them in the order that you want your cassette to be in. And so you'll just put them out in a line and put your pieces together, and that's what you're gonna do. Do we have any questions? If you have any, we'll be around, so just feel free to ask us if you need any help with anything.
So our scenario had to do with Roses Are Us Inc. And they're basically overall just trying to change the next generation of roses so that they were a different color and they would attract more customers, in the future. And so our promoter was the CMD promoter because we're working with a eukaryotic, like a plant, which was eukaryotic.
Okay, and then next, after the promoter, we had to use the binding site, and we used a 5
Prime cap.
Okay. We used cap and eukaryotic initiation factor because it said this binding site is used when dealing with eukaryotic organisms, which we had as the first thing.
Our next step was the coding sequence, and that was changing what trait of the rose we were changing. And we were changing the pigmentation of the rose, so we used the mill GP, which changed, actually we used the mill CP, which changed the color from rose red to blue. And that's the only trait we changed inside the rose. And next we used a stop codon to stop the transmission. And we had 3 options, and all 3 would work just as efficiently, so we went with UAG.
Then the very last thing is the terminator, which completely stops everything. So terminators that kill other people. So it says the ADH1 terminator is designed to work with eukaryotic organisms, and we had one, so we used the ADH1. It stopped everything. So this was our project.
Good job.
Our mission was to create a plant that fights pollution.
So we use the CMV plant promoter because obviously if we're creating a plant, it has to be a eukaryotic cell. And we used next the 5 cap and eukaryotic initiation factor binding site because this binding site is used when dealing with eukaryotic organisms, and a plant is definitely a eukaryotic organism. And then we use the start codon AUG because that is the only start codon.
For the coding sequence, we use cytochrome P450 because it breaks down pollutants in groundwater. And then we could have used any stop codon, so we used UGA. And finally, the terminator we used was ADH1 terminator because it's, again, for a eukaryotic organism.
Good job.
Yay.
So the purpose of our research is that we're going to find a fresh breeze that it can smell and glow in the dark.
Okay, so when we started, we did the Anderson collection for our one for prokaryotes again. Then we did AUG for our start codon. And we did the wintergreen odor for our scent. And then we did UAA for our stop codon. And then we did the RNPB1 terminator. And then we restarted for our second trait, the Anderson collection again for our promoter. For our binding site, we again did the ribosome binding site for eukaryotes. Our start codon was AUG, and we did a fluorescent pigment, which was the green firefly, so that it would glow. And then we did our stop codon, UGA, and then we did the double terminator.
Which one of your promoters is stronger?
Which one is stronger?
Yeah.
The second.
The second one? Did you want it to glow more than smell? Is that what you were thinking, or did you just kind of put them on there?
Well,
Okay. All right, good job. You guys want to go next?
So, Fresh Breeze Ink was basically trying to create a new human scent, and they also wanted it to glow in the dark. So we used the Anderson Collection 100 for our first promoter because out of Anderson 100 and 101, it's the stronger promoter. And so then we used the ribosome binding site to change the DNA to RNA, or RNA to DNA.
RNA to protein.
RNA to protein.
There you go.
And then we used the AUG because, you know, it works in all machines.
So these tell the ribosome what gene it has to read, which is wintergreen, and UPG is change the DNA from turning to RNA, and this one stop the transcription, which is from DNA turning to protein.
Since it also has to glow in the dark, we have to do like a new chain. Yes, and we use the other Anderson collection because it's the weakest one, and therefore we wanted the weakest gene to be the light glow in the dark. And then we started getting the ribosome binding site and the AGG to start the process. And then we select the green, and we use UGA to stop the translation and the double terminator to stop the transcription.
So you wanted it to smell more than it glowed.
Yes.
Okay, cool, good job.
So our mission was we had to make a bacteria that that you could put in meat that changes the color if it goes wrong.
so we used the promoter SBOA because it's upregulating in the presence of rotten meat. And then we used ribosome binding site because it's used with dealing with prokaryotic organisms. And then we put a starter codon and UG.
For our coding sequence, we use MOGP because it creates a green color that is visible to the eye. We use UAA for our stop codon, and then again we use RNPB-TI terminator because it's to work with a prokaryotic cell, which is bacteria.
So if there's rotten meat, what color did you see? Green? Green. Cool. Good job.